Ladies and gentlemen, welcome, and thank you for joining today's assessment of commercially available African swine fever virus real-time polymerase chain reaction assay webinar. Before we begin, please ensure that you have opened the WebEx participant and chat panels by using the associated icons located at the bottom of your screen. Please note all audio connections are muted at this time. You are welcome to submit written questions throughout the webinar, which will be addressed at the Q&A session of the webinar. To submit a written question, select all panelists from the drop-down menu in the chat panel, then enter your question in the message box provided and send. If you require technical assistance, please send a chat to the event producer. With that, I'll turn the webinar over to Liz Fernandez. Liz, please go ahead.
Good morning, everyone. I'm Liz Fernandez with the Professional Development Services Branch, and I'd also like to welcome you to today's Rubber Meets the Road webinar. Our speaker today is Kate Schumann. Kate is a microbiologist in the Proficiency and Validation Services section at the Foreign Animal Disease Diagnostic Laboratory at Plum Island. Kate has worked for the past 17 years on the validation of diagnostic assays for the detection of transboundary veterinary diseases of livestock, including foot and mouth disease, classical swine fever, and African swine fever. And with that, I'm going to turn the webinar over to Kate.
Thank you for the introduction, Liz, and welcome everyone to the webinar today from wherever you may be. I hope everyone is doing well. In fiscal year 2019, we applied and were awarded the Veterinary Services Grassroots microgrant to look at the assessment or evaluation of some commercially available real-time PCR assays for the detection of African swine fever virus. And the presentation today is the culmination of that work and a brief overview of our experience with them. I apologize. I know many of you on the webinar today, this will be repetitive, but I do strongly believe it's important to revisit what you're working with and understand the nuts and bolts so you're able to troubleshoot and better ask questions for any future work. So my takeaway from this is African swine fever is an extremely complex virus. It is the only member of its genus within the family Asfviridae. It's enveloped, in fact, it has multiple envelopes, and it has a double-stranded linear DNA genome. Its genome size is enormous. It's 170 to over 190 kilobase pair genomes. That makes it magnitude larger than many of the viruses that we work with here at Saddle. And with that large of a genome, we have many genes that are expressed, and there's been more than 150 open reading frames reported. This virus is also extremely large in size and is environmentally stable. When we hear African swine fever virus, most of us immediately go to the, oh, this is highly pathogenic, it's high virulence and high mortality rate. But in actuality, African swine fever virus and the different isolates and the different hosts exist in many different forms. So yes, while we are all familiar with that high mortality isolate, there's also isolates that cause mild disease all the way to those that only cause seroconversion without clinical signs. What I could find in the literature, and I'm happy to accept feedback from anyone on the call today, what I could find was there's currently 24 genotypes that have been recognized in the literature. The one that we are familiar with that's circulating in Europe as well as throughout Asia is that genotype 2 isolate. So we know that in the field, African swine fever is clinically indistinguishable from classical swine fever, and observations may include high fever, anorexia, weakness in the animals, bloody diarrhea may or may not be observed, petechia, and then, of course, upon necropsy, we see that classic enlarged spleen with African swine fever infection. So I'm largely a laboratory bench scientist with a healthy dose of paperwork mixed in, which I'm sure many of us can relate to. I'm also extremely detail-orientated, which lends to why I enjoy the work that I do so much. I've also been fortunate to be able to be on the ground in Georgia in 2007 with 3 other APHIS colleagues. That was during the outbreak event there. That really opened my eyes and was a pretty intense experience. I've also been able to spend a brief amount of time in Tanzania and work with some of the small farm holders there, as well as interact with slaughter slabs there during a very different type of African swine fever infection experience there. So that really brought me, I'm not sure if I'm full circle yet, but really helped me connect the dots on the importance of veterinary diagnostic laboratories, you know, not just at Battle, not across the United States, but really across the world. And that got me thinking, well, how important are pigs? And so I was thinking about, I wonder what animal protein source is the highest consumed across the globe. And so I took a look at the Pork Checkoff website, and if you haven't been there, it's in fact pretty interesting and more information than you could ever want to know about the pork industry, a lot of good information. And I was surprised to learn, you know, I really went in thinking everywhere I've traveled in the world, I can always find a Coca-Cola and I can always order chicken or find chicken. So I was really expecting chicken and or poultry to be the highest percentage consumed, but in fact it is pork. So when you couple that fact that approximately 40% of the world's meat consumption comes from pigs, and then you look at the map on the slide, which I pulled from the OIE website. It's from the World Animal Health Information Database. For those of you that haven't given that a try, I highly suggest going there. It's available to everyone, and it's, you can filter out for a number of different geographic locations as well as disease events, but I digress. If you couple the high pork consumption with the large amount of African swine fever events that have been reported just in the past 6 months, you can see what an impact, a socioeconomic impact, this disease has, not only on those of us that consume pork, but for all of the work that goes into its production, processing, and distribution. And if you're in a country that exports pork products, this will have an even larger impact on the economics and bottom line. So I did pull this graphic from the Pork Checkoff website. I know 4th of July is coming up, and many of you might be grilling some of these pork products as well as other items while you're socially distanced. But beyond the plate, I was really surprised to learn just how much of the animal is used, even beyond what goes out to the grocery stores. So there's a number of co-products or derived products coming from pigs. Those include pharmaceutical products as well as industrial products. Some things that might be more familiar to those of us include heart valves, as well as to those in the laboratory that are familiar with trypsin used in cell culture. On the industry side, pig skin is used for a number of downstream items, including clothing, gloves, shoes, as well as, footballs. And any meat products that are not destined for human consumption are used in commercial feeds as well as pet food. So if you couple pork production and an African swine fever outbreak that has high mortality, not only do you take a high-quality protein source off of people's plates, but you take all of these co-products or derived products out of the system, and that has impacts on the individual as well as families, commercial establishments, as well as everyone involved in its distribution and use. So back to the laboratory side of things, for African swine fever diagnostics, and this is by no means an exclusive or comprehensive list, But what we know based on the pathogenesis of the disease and where the virus is present in the highest titers or highest amounts, from live animals, we prefer to do diagnostics from whole blood. The animal is deceased, we request lymphoid tissues, mainly spleen, tonsil, as well as lymph nodes. I personally think the virus isolation for this virus is pretty cool, and it's unique in comparison to the other agents that we work with. It requires primary porcine cells, mainly monocyte-derived macrophages. And the image you see on this screen is from light microscopy, and those are ASF-infected monocyte/macrophages. And when red blood cells have been added, heme absorption occurs, and you see the classical rosette or circular formation there. For antibody and antigen detection, there's also PCR technology, both conventional and real-time, which we'll talk about today, antibody and antigen detection ELISAs, as well as immunofluorescence, immunoperoxidase, classical Sanger sequencing, as well as next-gen sequencing. So the objective, getting back to the objective of this study, so in 2007, there was a lot of attention brought to African swine fever virus, and then again in 2018 with the explosion across China, and now throughout Southeast Asia, there's really heightened preparedness, and a number of commercial vendors are providing diagnostics for African swine fever. So at the time we were writing the proposal, we went just out into the web and researched what was available, and these are the assays that we chose to evaluate. Many of them are going to sound familiar to you. They are strong in the world of veterinary diagnostics. So we evaluated assays from IDEX, IDVet, Inditel Biosciences, which purchased the veterinary portfolio from QIAGEN. And then we also looked at assays from Tetracore as well as Thermo Fisher Scientific. So why would we even want to take a look at commercially available assays? You know, currently, and for well over 10 years now, we've had the assay that we use at FATL and the assay that is deployed in the National Animal Health Laboratory Network. It was developed by our colleagues in the Agricultural Research Services side, and that's worked well for us for the purposes that we have needed it for. But again, as I mentioned, with this heightened preparedness, we're often asked by stakeholders, both in industry and both global collaborators, oh, well, have you heard of this diagnostic assay? What do you think about this? And really, I'm a strong believer in I can provide you my opinion, I can provide you my theories, but nothing means as much as the hard currency of generating data. So a couple of reasons to take a look at commercial assays in addition to what I'll call the reference assay, which we use at FATL and in the state veterinary diagnostic laboratories, are for the reference assay, we purchase each component individually. And when I say each component, I'm talking about enzymes, primers and probes, internal control, as well as a detection system for that control. With a commercial assay, and I'll go over it in detail on the next slide, the majority of them come ready to use right out of the box. For the reference assay, we are required to perform a set of quality control on the primers and probes to ensure they are working as we intend them to. And with a commercial assay, we would expect that to already have been completed. For my next qualifier, the operator skill set, it took me a little while to tease that one out because really I think that if you have a molecular background, whether you're using a reference assay or the commercial assay, they're pretty easy to use. But if you're training someone new, someone who's never done molecular work before, there's just less tubes, less things to keep track of with the commercial assays. Usually they're just one or two tubes, and I kind of relate that back to, you know, I'm not very good in the kitchen, I can make something out of a box a lot easier than I can making it with individual ingredients. So that's how I think about that qualifier. And then again, for the reference assay, our technical support is going to come from our peers and colleagues, which is wonderful. But in addition to that, when you're using a commercial assay, you have that technical support base from the vendor. A couple of things we looked at with the assays was the cost per reaction, and I want to add that on the next slide when I go over the cost, that is what we paid at one time point. The cost can fluctuate based on what kind of entity you are, as well as how much of the assays you are buying. We looked at what controls were included, including endogenous, so things such as beta-actin or GAP-DH to assess sample quality. Was there exogenous control and detection system included, and that's usually added to help qualify and ensure the process was carried out appropriately. And then did these kits or commercial assays include an African swine fever positive amplification control? We also will briefly mention what our experience was with the vendor in terms of ordering, any technical support if we needed it, as well as the time frame from when we ordered it to receipt of the product. Another layer of using these assays, where only one shipped and generated in the United States. So on Plum Island, in order to receive diagnostic assays from overseas, we're required to complete a risk assessment, and then we request an import permit to receive those assays. So while it's not difficult, it is another layer of paperwork added on to those assays that come from overseas. And only the assay from Tetracore, which is located in Maryland, this was not required for. The other assays come from United Kingdom, Netherlands, France, and Germany. And the order of arrival, surprisingly, we received the one from the United Kingdom, Thermo Fisher first, followed by Indical, IDVet, IDEX, and the one from the United States last. In terms of cost, Thermo Fisher was the most cost-effective, closely followed by the assay from IDEX. So this slide really gets into the weeds and details of the diagnostic assay, but for those of us that work at the laboratory bench and when we're thinking about the fitness for purpose of the assays, these may or may not be important. So we calculated the runtime based on the denaturation and elongation steps. We did not include the heating or cooling ramp times, as those can be variable. So the quickest runtimes were from IDVet when run in fast mode, as well as the reference assay. All their assays ran approximately about 1 hour in time. All of the commercial assays came with enzyme included, with the exception of the IDEX assay, which could be purchased separately. Endogenous control detection was included in the IDEX, IDVet, as well as the Indical Bioscience assays. Exogenous control and the detection system for that was not included with the IDEX assay, but could be purchased separately. IDVet did not provide that, but the other three vendors did. Moving along to the positive extraction control, that can be a little bit confusing. Historically, we've included a disease target-specific extraction control, and we've since moved away from that and used our internal controls as our extraction control, and all vendors provided some type of positive extraction control. The only one that provided disease-specific positive extraction control was IDVet. And then for the African swine fever specific positive amplification control, all vendors provided that in the kit with the exception of IDEX, and that could be purchased separately. So the first analysis we did with the different assays was inclusivity testing, and that's just the word for meaning how many different isolates can we throw at the assays, and do all assays detect all isolates. So with the help of our repository on island, we identified 26 different isolates. We grew them up on macrophages and titered them. And as we expected, all assays detected all 26 isolates that were tested. The earliest CT values that were produced from this, and we thought the assays that were easiest to use, had the least amount of components that needed to be added together, were IDVET and the Indical 2.0 assay. So there was an initial Indical assay that had some performance issues, and then communication with the company informed us they were improving that assay, and we were able to obtain the beta test of the next version of the assay, and it worked better than that initial test. So this is a very busy slide. I apologize, but I wanted to use it as a demonstration of all the data that is collected just for inclusivity testing. So these are plots of the cycle thresholds versus the assays that were used for 3 independent replicates for each isolate that was used. Next, we moved into analytical sensitivity testing, or looking at a comparison of how many viral particles or to what dilution can these assays detect ASF. We chose 4 isolates, Georgia, of course, being the one that's currently circulating in Europe and Asia. We chose a Rome isolate, a Tangani isolate, which is from Malawi, an isolate from Zaire, and Zaire is now known as the Democratic Republic of the Congo, just to provide some context to where that isolate is from. So dilution series of those isolates are made in an appropriate sample matrix. We made them in 10% spleen homogeneous, and then we ran 3 independent replicates with each isolate. So again, this is a very busy slide, again, showing all of the data points that were collected, plotting cycle threshold versus the dilutions that were made of each isolate and run against each assay with each independent run. And then a little easier to quantify that, when you compare, our metric is, a cutoff point is the last dilution where all 3 of the 3 independent replicates result in a positive reaction. So with that criteria and using these 4 isolates, the assay that performed the best, the Thermo Fisher scientific assay, and close behind was the assay from Tetracore. And if you remember on a previous slide for inclusivity testing, where I talked about the IDVET and the Indical bioscience assays producing early CT values, and often I hear, or we hear people talking, oh, look at these early CT values. This assay is performing better than other assays, or it's more sensitive. Well, this is a perfect example of where that does not hold true. An earlier CT value at any dilution does not necessarily translate to greater sensitivity. As you can see here, the IDVET assay was a log-less detection across isolates in comparison to the Thermo Fisher scientific assay. So conclusions from this brief project were that, as we expected, each assay was able to detect all isolates in the inclusivity testing portion. The best performers with the 4 isolates that we used was the Thermo Fisher scientific and Tetracore assays. And between those 2 kits that performed well in analytical sensitivity testing, the Thermo Fisher was the most cost-effective. And while we found the IDVET assay to be user-friendly, low-cost, and produce those early CT values, its analytical sensitivity was limited compared to others. And then my last point is where I'll get on my validation soapbox a little. I know we often think about or are told, oh, this assay is validated, and might be told that by another laboratory, a commercial vendor. And I just urge all of us to go back and ask, well, how was it validated? the data I have here today are important puzzle pieces to each of these assays, but there are definitely gaps which may or may not impact how these assays could be used in the field. It's all about fitness for purpose. So our study did not cover the 24 genotypes. It did not, while we don't expect there to be issues with exclusivity testing or looking at specificity, we did not generate data to prove that. Another gap is repeatability. What could be addressed is inter and intra-assay repeatability, as well as inter and intra-laboratory repeatability. Diagnostic specificity is also a gap. For the OIE validation criteria, it is important to look at the assay you want to use versus the population you're going to use them with. Using this assay on pigs from East Africa is going to look very different than using these assays on pigs from the United States. And of course, something I always hope for is diagnostic sensitivity, is obtaining naturally infected field samples, and I know those are difficult to get, but if anyone has connections or suggestions where we might be able to collaborate with international partners on evaluation of field samples, we are always open to that. Okay, with that, I'd like to thank the Veterinary Services Grassroots Committee for the funding of this project. I'd like to thank Leslie Blakemore, who performed the majority of the bench work for this project. Thank you to Dr. Vivian O'Donnell for getting us off the ground with the macrophage work, for growing those 26 isolates, as well as Phil Doucette for working with us with those macrophages. Also in agricultural research services, I'd like to thank Sarah Pruitt, Dr. Elizabeth Ramirez for all their hands-on work, and their supervisor for supporting them, Dr. Manuel Borca. I'd like to thank the donor pigs for the material that we used to generate macrophages. And as always, Dr. Monica Rising with the Center of Veterinary Biologics. And with that, I've completed my talk, and I'm happy to take any questions or accept any feedback anyone may have for me. Thank you.
Okay, we just have a couple questions in the chat. The first one is, was the cost of the extra items that need to be purchased to run the assay included in the cost analysis?
Yes, they were.
Okay. And the next one is, what thermocyclers are used in these assays? Are they different for different kits or the same?
So that's a great question. So we use the ABI 7500, and we followed the manufacturer's cycling parameters. So while I really am interested in moving on to maybe a newer generation of thermocyclers, I find that I think it was in 2007, 2008, where the avian influenza outbreak across the globe, you can almost find a 7500 in a number of laboratories across the world. So yeah, that's where we did evaluate it, and for that reason, just the accessibility of the 7500 real-time PCR platform seems to be the greatest globally.
The next question is, have you worked with FAO in the past, and because they may be able to collaborate internationally?
We have worked with the FAO in the past, but for other transboundary animal diseases, mainly foot and mouth disease virus. But yes, we can definitely reach out to the FAO.
The next question is this something we would want the National Veterinary Stockpile to consider, or the lab to have contracts to be ready to use these if we, unfortunately, if we got ASF?
You know, that question is probably beyond me politically, but in my opinion, yeah, I definitely think reaching out to some vendors and understanding what the supply chain could be for the United States as well as globally is going to be important. I know some of these vendors are currently supplying China, and it would be interesting to hold conversations from both stakeholders' point of views to see what's worked well and what hasn't worked well.
Okay. With your ASF international perspective, as well as the results from this study, how do you foresee the diagnostics evolving?
That's a great question. So I think with African swine fever, there's this range of backyard producers all the way to these massive commercial industries. So the needs from those are going to be different. I know I haven't evaluated them, but I'd like to get my hands on and take a look at what might be a little more cost-effective and user-friendly, the LAMP assays for African swine fever. But I do really think I'm a big fan of real-time PCR for its sensitivity, specificity, and really the time frame that it can be run in. I think it's a very useful tool for the presence-absence purpose.
Okay. The next question is for field samples. Are you willing to do the testing internationally, or would the testing have to be conducted at Plum?
I prefer to do the testing internationally. I prefer collaborations, and I really enjoy working in country. It would also be greatly appreciated if we could have some to build up the repository of field samples here for African swine fever virus. What we have is just very low quantities in terms of what can be used. And when we talk about doing repetitive studies to ensure the statistical, I don't know if power is quite the right word, but assurance in your results, we really need a sufficient quantity. But yeah, I'm all for doing collaborative work in country. We'll also see the benefit of being able to bring some material back. So I guess both.
Okay. You have a lot of great comments, Kate, about what a great presentation this was.
Thank you.
The next question is, any information available from the different kits in terms of the regions targeted on the genome?
So we did ask, and we don't know exact genome position, but we do know they all are located in VP72.
Okay. What is the cost of your in-house assay compared to the kits evaluated?
Sure, great question. And that's a little bit hard to determine based on the volume of primers and probes that we order. But so let's say the Thermo Fisher Scientific assay, I think, was about $7.30. If you break down the components for the in-house assay, that you're going to put that around, and this is a median, around $3.50.
Okay. Did you find the grassroots funding concept particularly useful for small research projects like this one?
Yeah, absolutely. It really, we were excited to receive it and get off the ground. And with the money we were able to use for this project, we got approval from our management to continue the work with analytical sensitivity testing. So it's really nice to be able to kind of dip our toes into, okay, are these assays worth looking into further, kind of gave us that starting point or the springboard.
That's great. So we have another comment, too, from Aruna from the NCFAD Canada, and they said that they would be happy to collaborate with you to do an interlaboratory comparison. They have a large collection of positive clinical samples from Africa from naturally infected animals. So that's great.
Thank you, Aruna.
And like I said, Kate, you have a lot of comments thanking you for this great webinar. Do we have any verbal questions in the queue?
So I Ladies and gentlemen, if you would like to ask a question via phone, please press #2 on your telephone keypad to be entered into the question queue. Once again, pressing #2 will enter you into the verbal question queue.
Okay. We'll give everybody just a couple minutes or so. See if we get any more written questions. Here's another one. What other virus would you suggest a similar project be done for?
Well, this is my opinion. I think classical swine fever virus really deserves some attention. I know we are all at heightened awareness and preparedness for African swine fever, but classical swine fever is right next door in Dominican Republic, and I would really like to take a look at that as well.
We have another question. It says, from which ASF genotype the 26 isolates tested were coming from?
That is an excellent question, and we do not know all of the genotypes of all of those 26 isolates, but we are working on it. We do know we definitely included the genotype 2 with the Georgia isolate. That's a great question.
Okay. We have another question about will there be any more webinars? Well, we do have a lot more webinars coming up. I'm not sure if they mean about this project or other topics. If they want to write to me and let me know, that would be great. And then we also have a question that says, please provide the panelist questions on the speakers, the questions. And thank you for this presentation. They want to know if there's going to be any more webinars on this project.
No, that's a great question. Right now, we're working on, we just got out the door FMD Seneca work, and right now we're really deep into rabbit hemorrhagic disease, and once that passes, I would hope we could be able to revisit these commercial assays and address some of the gaps.
Okay, here's another question. Environmental sampling is difficult to obtain. Do you have a recommendation on how a person in the field would take an environmental sample? And if so, how much sample needs to be sent to the lab in order to extract ASF?
Sure, that's a great question. And we actually did this in Tanzania. We wanted to take a look at between pens if we could find the virus on things such as the pen door handles, the feed troughs, the water troughs, et cetera. So it's really actually fairly simple, and there's probably multiple ways to take environmental samples. We simply use a Dacron swab, swab back and forth with a good contact on those different surfaces, and then placed it in like a PBS or MEM media. And the minimum amount that we need in the laboratory is around 250 microliters. There's always that trade-off between having enough volume versus diluting out what you might be able to detect. So usually when I've been in the field and this isn't right and this isn't wrong, we usually use about 1 ml of diluent, and that provides enough material for the laboratory.
Okay. The next question is, where should we look for in-depth considerations of routine domestic ASF diagnostic surveillance?
That is a good question, and I know there is, I have to go back to the NULM program office, but there is an excellent manual on integrated surveillance for ASF and CSF, and I'm not sure who asked the question, but Liz, can you see who asked the question?
Yes, and I can send you all these questions. So
okay, perfect. Perfect. I'd be happy to address that more specifically when I have the correct information.
Yeah, and we'll have everybody's email too. We get a report on who signed on. So
okay, thank you.
I can get all that information. Yeah. Do we have any additional questions?
There are currently no questions in the verbal queue, but as a reminder, ladies and gentlemen, #2 will enter you into the verbal question queue.
Well, if there aren't any more questions, I want to thank Kate for presenting today. This is a great project and great presentation. I just want to let everyone know that's also on the line, the National Training and Exercise Program will be hosting a webinar on July 23rd on planning and implementing a carcass disposal site in Minnesota. That information will be sent out within the next week. And be sure to watch your emails for additional webinars coming up in the next month. And with that, I will wish everyone a great afternoon. Thanks for joining.
Thank you.
That concludes our conference. Thank you for using Event Services. You may now disconnect.